गुड मॉर्निंग फ्रेंड्स। आवाज येतोय? ऑडिओ व्हिडिओ क्लिअर? ओके, क्लिअर. चलो, very good morning and welcome to Unacademy. This is Abhijit and today we are going to discuss about the CAM pathway. Okay, so in last lecture we already discussed the Kelvin cycle, then C4 pathway. Okay, Kelvin cycle is also called as C3 pathway. And in the C4 pathway, we already discussed the photorespiration. Okay, so the photorespiration, if you see what is coming out from the photorespiration is uh, the glyoxylate. Okay, so the glycolate. So glycolate, if you see, it is the two carbon atoms. So the photorespiration is also called as the C2 compound. Okay, so the C2 because of C2 compound, it is also called as the C2 cycle. Okay, so we already discussed the C3, C4 and C2 cycle. So the today we are going to discuss about the CAM pathway. So this pathway is only present inside of the desert plant. So it is very beautiful modification, or you can say the very the intelligent decision by the environment. Okay, so I always say that nature is very smart. Okay, so this is the one of the smart decision taken by the evolution in the course. Okay, so this plant can photosynthesis in the presence of very high heat. Okay, so how they do that is the topic of today. Okay, so before the starting lecture, so guys, this is the Crack Maharashtra CET channel. It is for the batch 2021. Okay, so the name of batch is Target Top 100 Ranks MHT CET 2021. If you do not have subscribed the channel, please subscribe it. This is Abhijit Madkar. I have more than 6 years of experience in the field of biology and also cracked NET CSIR with All India Rank 60 in life science. Okay, so we are here on the India's largest platform. If you want to enjoy more about the education with Unacademy, so it is very necessary to download this app. This app is available on the App Store and the link is given in the description. Okay, मित्रांनो, या व्हिडिओच्या डिस्क्रिप्शन मध्ये तुम्हाला लिंक दिलेली आहे या ऍपची. तुम्ही तिथून सुद्धा याला डाउनलोड करू शकता. हे टेलिग्राम ऍप आहे. या ऍप वरती तुम्हाला एमसीक्यूज आणि नोट्स अवेलेबल आहेत. ओके, सगळ्या विषयांच्या आणि एमसीक्यूज पण अवेलेबल आहेत. आणि या ठिकाणी तुम्हाला डेली अपडेट्स आणि त्याची जी काही लेक्चरचे लिंक्स वगैरे आहेत तर त्या सुद्धा तुम्हाला इथे अवेलेबल होतील. ठीक आहे मित्रांनो, हे आहे आपलं अनअकॅडमीने तुमच्यासाठी काही special facilities आणलेले आहेत. So these are the facilities for you. So guys, the 12-month subscription, if you see, the fee is 14,000 and for 24-month fee is 21,000. Okay, so if you use this code the capital Abhijit, it will definitely give you the 10% discount in this fee. Okay, so this fee will give you the subscription will give you the offline facility. So you can download the lecture and you can enjoy it as per your time. Very good morning guys, very good morning. Okay, then the MCQs practice, then doubt solving session, dedicated doubt solving session. तुम्ही तुमच्या आवडते एज्युकेटर सोबत इंटरॅक्ट सुद्धा होऊ शकता या ऍप मधून. ठीक आहे, तर या सगळ्या फॅसिलिटीज आणि तुम्हाला डिस्क्रिप्शन मध्ये मी सगळी इन्फॉर्मेशन याची दिलेली आहे. तुम्ही एकदा पाहून घ्या. ते सर्व गोष्टी तुम्हाला सबस्क्रिप्शन मध्ये अवेलेबल आहे. ओके, सो थँक्यू व्हेरी मच अँड लेट्स स्टार्ट द लेक्चर. सो the CAM pathway. Okay, so this is the CO2 fixation in a desert plant. The CAM pathway is a way sorry, this is the way to fix the CO2. Okay, so this is the way to fix CO2 for the desert plant. So it is only inside of a desert plant. So why desert plant required the different pathway? The very important thing, guys. Okay. So if you see in a C3 pathway and a C4 pathway. Okay. So this is the Kelvin cycle. This is your Kelvin cycle, and it is present the C4 plants. So these plants are found in a tropic and subtropic region. Tropic and subtropic. Okay, so these plants like sugarcane, maize, this plant having its special feature is a cran's anatomy. Okay, but your cam do not require cran's anatomy, and they only found in a desert. Okay, so there is actually the different the method in a CAM pathway for the fixation of a CO2. So the CO2 is fixed. So what is the problem actually in the fixation of a CO2? So guys, for example, if you see the structure of a leaf, for example, I'm going to cut the leaf. Okay, so let's take one leaf. Okay, so if I cut it from here, and if you see the this portion, cuts portion, so it will looks like this. Okay, so here is the one term layer, and the cells are present here. Okay, so guys, what I want to show you is this part. If you see, here is a gap between the cells. Okay, so this gap is nothing but the stomata. This gap is nothing but the stomata. You can say it is the nose of the plant. ओके तुम्ही याला प्लांटचं नाक म्हणू शकता रेस्पिरेशन साठी. CO2 आतमध्ये घेता येतो आणि O2 बाहेर सोडतो. ओके, दोन्ही गोष्टी ऍट अ टाइम पण होऊ शकतात. CO2 आतमध्ये, O2 बाहेर. ठीक आहे, तर या ठिकाणाहून ज्यावेळेस हे ओपन होतं ना, याला स्टोमॅटा म्हणतो आपण. याला काय म्हणतो आपण? स्टोमॅटा. ज्यावेळेस टोमॅटो ओपन होतो ना मित्रांनो, त्यावेळेस CO2 आतमध्ये जाताना जर हीट जास्त असेल तर पाणी बाहेर येतं वेपर फॉर्म मध्ये. ओके, गॅसेस फॉर्म मध्ये किंवा वेपर फॉर्म मध्ये. मग हे पाणी जर बाहेर आलं तर तुम्ही जर विचार केला तर C3 केल्विन प्लांट मध्ये जर तुम्ही विचार केला तर 1 ग्रॅम कार्बन डायऑक्साइड फिक्स करण्यासाठी जवळजवळ 400 to 500 ग्रॅम पाणी बाहेर येतं. Okay, so they required 400 to 500, I mean they lost 400 to 500 gram of water for the 1 gram of carbon dioxide fixation. Okay, and these plants require 100 to 200, about 200. Okay, and this your CAM plant required only 20-30. Okay, so the if you see the efficient way to fix the carbon dioxide is nothing but the CAM pathway. Okay, so this pathway is very the efficient if you say in a desert plant. So if you think the C3 and C4 pathways which are present in a desert plant, so desert plant will they will not, I mean it is not easy to live inside of a laser inside of a desert with this the C3 and C4 plant. Okay, so that's why the CAM pathway is very important. This pathway is actually found in the family of the desert plant, and that family is called as the Crucilacean family. Okay, so the Crucilaceae family is there. Crucilaceae. Okay, so this is the family of plant, and that's why I underline as per your rules of the a botanical name. Okay, so the botanical name or you can say the binomial nomenclature. So that's why I made an underline here. It's a Crassulaceae family, which is the family of a desert plant. Family of desert plant. So this family had the different way for the carbon dioxide fixation. So this CO2 fixation way is called as the cruciation acid metabolism. Okay, so the logic behind acid I am going to explain in this the lecture. Okay, so don't worry, it is similar to the C4 pathway by many aspects but differ in a few important feature. Okay, so the similarity is with the C4 pathway. Okay, so the similar with the C4 pathway but not the complete similar. Okay, so there is a few differences as compared to the C4 pathway. So the camp plants lack Kranz anatomy. So the biggest difference, C4 plant shows the Kranz anatomy, but your camp plant never shows the Kranz anatomy. Okay, so Kranz anatomy is absent in a C4, sorry, in a CAM plant. PEP carboxylase and Rubisco are present in a mesophyll cell. Okay, so we know that in a C4, the the picture is a different. Rubisco is present in a bundle seed cell, and your PEP carboxylase is present inside of the mesophyll cell, outside part. Okay, so the mesophyll cell here in the CAM pathway, PEP carboxylate and Rubisco both are present in a mesophyll cell. Okay. Can open the stomata at a cold desert and close during a hot, a dry day. This minimizes the loss of water. Okay, so the cam opens stomata at a cold desert time means if you see as compared to the C4 pathway, the stomata are required for the breathing. Okay, so it is open at night and close at डे ओके एखादी गोष्ट नाईटला काम करत असेल ना मित्रांनो तर आपण त्याला मोस्टली स्कोटॉपिक किंवा असं काहीतरी नाव देतो स्कोटो म्हणतो आपण नाही का आपण आपल्याला विजनला पण नाही का रॉड सेल जर तुमच्या रात्री काम करतात रॉड सेल आपल्या डीम लाईटला काम करतात आपण त्याला काय म्हणतो रे स्कोटॉपिकच म्हणतो ना बरोबर ते का म्हणलं जातं ते स्कोटो वर्ड मोस्टली आपण नाईट साठी वापरतो पण यांना आपण स्कोटो ऍक्टिव्ह म्हणतो ओके स्कोटो ऍक्टिव्ह म्हणजे ऍक्टिव्ह ऍट नाईट ऍक्टिव्ह ऍट नाईट किंवा डिम लाईटला म्हणतो आपण ठीक आहे ऍक्टिव्ह ऍट नाईट किंवा डिम लाईट अँड स्टोमॅटा इज अ क्लोज ॲट द डे टाइमला क्लोज असतो कारण डेटला काय आहे मित्रांनो तर लाईट इंटेन्सिटी खूप जास्त आहे आणि दुसरी गोष्ट असते टेम्परेचर actually the reason behind the closing is the temperature okay so this temperature is a very high if stomata is open so the lot of water will leave the plant okay stomata open झाला की water निघून जाईल plant मधनं आणि असं काय होऊ नये म्हणून गरजेचं आहे की स्टोमॅटा दिवसा बंद झाला पाहिजे आणि रात्री ओपन झाला की रात्रीच्याला ओपन होतोय म्हणजे काय होणार आहे रे रात्री CO2 आतमध्ये येणार आहे so CO2 will coming inside of a cell at a night time and daytime it will convert CO2 into the sugar okay so the how this happen in a different timing okay so this is the actually the beauty of this CAM pathway so that more than two things are happening in a different things I mean different the uh, timing okay so the CAM CO2 is assimilated at a night and reduced during the day in the presence of light. Okay, so CO2 is assimilated at night means at night it is coming inside of the plant and during a day it is used by the plant. Okay, in the presence of light. So this is the CAM pathway, guys. Let's have a look. पाहून घ्या एकदा. कोणाचं काही क्वेश्चन आहेत का मित्रांनो? क्वेश्चन असतील तर विचारा. आहेत का कोणाचे काही क्वेश्चन? चलो आपण काय करूया मग आता, याला व्यवस्थित समजून घेण्याचा प्रयत्न करूया तुमच्या कॅम्प पाथवेला. ठीक आहे. आता कॅम्प पाथवे मध्ये बघा काय होतंय. तर या ठिकाणी तुम्हाला दोन कंडिशन्स ऑलरेडी दिलेल्या आहेत. तर ही तुमचे नाईट वाली कंडिशन आहे. सो दिस इज अ नाईट and this is a day. Okay, so the highlight is present in the daytime. So in the night, CO2 is going inside of the cell or inside of a plant, so it is combined with the H2O. We know that the anhydrase enzyme which convert it HCO3 plus H plus. Okay, so actually it is converted into the H2CO3, but H2CO3 is a very unstable compound and automatically convert into the HCO3 plus H plus. Okay, so the PEP phosphoenol pyruvate, and this is the PEP carboxylase. Phosphoenol pyruvate carboxylase, the very important enzyme as compared to the C4 pathway and the CAM pathway. Okay, so this enzyme is actually the part which is going to fix this carbon dioxide is CO3. Okay, this carbon dioxide on the PEP phosphoenol pyruvate. So this phosphoenol pyruvate is a C3 compound, and when your CO3 is fixed with the PEP, so it will convert itself into the C4 compound. Okay, and oxaloacetic acid, that is OAA, is present now inside of a cell. This oxaloacetic acid is immediately converted into the malate, that is malate dehydrogenase enzyme, where the NADH is used by the malate, and this malate, or the you can say the malic acid, can be stored inside of the a vacuole. Okay, so this malic acid is stored inside of the vacuole. Okay, so now we have the malic acid inside of a vacuole, and it required each and every time when the malate is coming outside inside of the vacuole, so that time we required the H plus to convert it malate into the malic acid, and this pumping of H plus required the ATP. Okay, so actually the ATP is used here, and your reducing power is used here. Okay, and here is also we require the energy PEP carboxylates. Okay, so now in the night time, if you see, we have a lot of malic acid. In the night time, what happened, we have a lot of malic acid. रात्रीच्या काय झालं, malic acid जमा झाले. So malic acid, so the pH is going to be a decrease. pH is a decrease. So the process is called as acidification. Okay, so it is called as acidification. Let's say we will have the written slide after this slide. Okay, so don't waste time on the acidification process. So here in the daytime, if you say the stomata is open first of all, so the nothing is going inside. Actually stomata is not the closed. Okay, stomata is not closed for the entry of a CO2, but it is closed because the water can't leave. Otherwise H2O will live in the form of a gas, that is vapor. Okay, so here when the CO2 is coming inside, so that time H2 is also moving out of the cell. Okay, and to avoid this loss in a daytime, if you see the daytime temperature is very high, and that's why the loss is also very high. So to avoid this loss, stomata is completely closed at the daytime. So in the daytime, in the presence of a light, so light is coming in a daytime, we know that the light is present. So in the presence of a light, this H plus is coming out of the vacuoles. Okay, so let's see, this H plus is coming out of the vacuole. So the malic acid is converted into the malate, and this H plus is coming out. So the if you see the H plus concentration is moving, and that time your malate is also coming out of the cell, and this malate is converted now into the oxaloacetate. So here the NADH is NAD is used, and NADH is a form. Okay, so guys, let's see, either malate is converted into the pyruvate, or the malate is converted into the oxaloacetic acid, and then oxaloacetic to the PEP. So this PEP is a C3 compound, and your pyruvate is also C3 compound. दोघं पण C3 आहेत मित्रांनो. हा C4 आहे malate, आणि हा पण C4 आहे. यामधून एक कार्बन काढून घेतला, यामधून एक कार्बन काढून घेतला malate मधनं. पॅलेट मधनं डायरेक्ट पण कार्बन काढून घेता येतो. डायरेक्ट कार्बन काढून घेतला तर पायरुवेट मध्ये कन्वर्ट होतं, आणि जर इनडायरेक्टली कार्बन काढून घेतला वाया ऑक्झॅलॅसिटिक ऍसिड तर पीईपी मध्ये कन्वर्ट होतो. सो दिस बोथ कार्बन डायऑक्साइड which is the extracted from the oxaloacetate and the malate, they are going to enter into the Kelvin cycle. So this is your Kelvin cycle. Okay, and after the Kelvin cycle we know that the actual result is the sugar. Okay, so the it is for the sugar. So this is actually the pathway. Daytime it is behaving like the different, and night time it is behaving like a different. So night time the assimilation of carbon dioxide is happen, and the daytime the actually the carbon dioxide is used and converted itself into the sugar. Okay, काय होतंय मी परत एकदा सांगतो तुम्हाला एकदम शॉर्ट मध्ये 2-4 लाईन मध्ये. मित्रांनो, खूप जास्त हीट असल्यामुळे डेझर्ट अँड प्लांट मध्ये तिथे खूप जास्त हीट असल्यामुळे जर कार्बन डायऑक्साइड आतमध्ये घेण्यासाठी स्टोमॅटा ओपन केला कधी डे टायमिंगला, तर काय होणार आहे, H2O बाहेर जाईल, आणि H2O खूप जास्त बाहेर जाईल. एक तर पाणी कमी आहे, त्यात जर लॉस झाला तर चालेल का, नाही. म्हणून काय करायचंय, तर रात्रीच्याला नाईटला CO2 आतमध्ये घ्यायचा. ओके, ज्यावेळेस नाईटला काय असतं रे, टेम्परेचर कमी असतं, कूल नाईट असतात, डेझर्ट नाईट कूल असतात. ठीक आहे, मग टेम्परेचर कमी असल्यावरती H2O बाहेर जात नाही, CO2 असिमिलेट झाला, CO2 मॅलेटच्या फॉर्म मध्ये स्टोअर करून ठेवला, मॅलिक ऍसिडच्या फॉर्म मध्ये व्हॅक्युअल मध्ये. आणि ज्या वेळेस पण सनलाइट आतमध्ये येते त्यावेळेस काय होतं की इन द प्रेझेन्स ऑफ सनलाइट भरपूर सगळ्या प्रोसेस ऍक्टिव्हेट होतात. त्यातली एक इम्पॉर्टंट प्रोसेस आहे की पहिला स्टोमॅटा क्लोज होतो इथला. ओके, दुसरी गोष्ट इथं मॅलिक ऍसिड टू मॅलेट कन्व्हर्जन स्टार्ट होतं, आणि malate बाहेर येऊन either it will enter into the pyruvate or the oxaloacetate and then PEP. Okay, so these are the C3 compounds and malate is a C4. So the one carbon dioxide is coming from the oxaloacetate to PEP, and one is coming from the malate to pyruvate. Okay, so the two carbon dioxide enter into the Kelvin cycle, and the Kelvin cycle results into the formation of a sugar. Okay, so event in a camp. Formation of malic acid during night is called as acidification. Okay, so acidification means the formation of a malic acid during a night. So in night we know the CO2 is fixed, and it is added on the PEP phosphoenol pyruvate, which is converted itself into the malic acid, and which is stored inside of the vacuole. Okay, so malate is stored in the vacuole during a night. So the two things which is happening during a night, in the presence of a light, so it is a night things. In the presence of a light, a malate is released from the vacuole and consumed by the C3 pathway consumed by C3 pathway by same cell and called detoxification. So the first phase, this is the first phase, guys. So I mistakenly written it a second. So this is one first phase. Okay, so this is phase 1 and this is phase 2. So the phase 1 is acidification. Phase 1 is acidification, and phase 2 is the de-acidification, actually, not detoxification. Okay, so it's a de-acidification. Okay, deacidification. Okay, an example of pathway, the CAM pathway plants are the aloe. Okay, we know that it is the alveolar like the plant. Opuntia, we know this plant is because the image of this plant is present in our uh, textbook. Okay, so significance of a CAM pathway. So the CAM mechanism enable plants to maximize their water efficiency or water use efficiency due to the scotoactive stomata. Stomata is not open at the daytime, so the water loss is minimized. So CAM loses 50 to 100 gram of water for the 1 gram of carbon dioxide. Okay, so the 1 gram carbon dioxide fixation are required 50 to 100 gram of water into the your CAM pathway, CAM plant. Okay, and 250 to 300 gram in C4 and 400 to 500 in C3. If you see the efficiency, so the water use efficiency is very high in a CAM pathway as compared to the C3 and the C4. C3 and the C4 required very the large amount of water for the carbon dioxide fixation. Elevated internal concentration of a CO2 suppress the photophosphorylation. Okay, so the carbon dioxide is a very high inside of a cell. If you see the cells of the CAM pathway, the carbon dioxide is very high. Okay, so the high concentration of a carbon dioxide does not permit the oxygenase activity of rubisco. Okay, so this activity of rubisco is not permitted due to the elevated level of carbon dioxide. So this is the very good the significance of a CAM pathway, and thus these plants are specially adapted to the arid environment, or you can say the desert environment. Okay, so arid or a desert environment or the arid or desert plants are good in a CAM pathway. So the factor affecting the photosynthesis. Okay, so this is the very important point. So we are now not talking about the only the CAM pathway, we are talking about the photosynthesis. Okay, so the each and every the light reaction and the whole type of a dark reaction we are going to discuss here. Okay, so the first thing is the light. We know the photosynthesis is completely depending upon the light. Quality and the intensity of light can affect the photosynthesis. Okay, quality and the intensity. Quality म्हणजे काय तर आपल्याला माहिती आहे की photosynthesis ला different regions लागतात. So the red light shows the high rate of a photosynthesis before the blue light. Okay, so the blue light is the second. म्हणजे photosynthesis कुठे कुठे होतं रे, जर तुम्ही असा 300 to 700 चा region घेतला तर सुरुवातीला फोटोसिंथेसिस हाय असणार आहे आणि शेवटी हाय असणार आहे. ओके, सो द photosynthesis happening inside of a blue and a red region. ओके, ब्ल्यू अँड रेड रिजन, याच्यामध्ये बाकी 2-3 ठिकाणी फोटोसिंथेसिस कमी होतं. सो द photosynthesis also required quality of light and intensity. So the intensity increase in intensity is result into the increase in photosynthesis. So it is directly proportional to the photosynthesis process. Carbon dioxide, it is also the factor which is the maybe the rate limiting. So the 0.03% of carbon dioxide is present in environment. तुमच्या बुक मध्ये चुकून 0.3 झालेला आहे, 0.3% असं झालं आहे, 0.03% present in environment. Increase in carbon dioxide increase the rate of a photosynthesis. So the if you say the carbon dioxide present which is present in an environment, if you increase the concentration of carbon dioxide, it will result into the increase in the rate of a photosynthesis. Okay, मग आणखी 1% ने जर कार्बन डायऑक्साइड वाढला तर रेट photosynthetic rate वाढतो. म्हणजे फॉर एक्झाम्पल Now we are having 0.03%, for example 0.045%. तर इथपर्यंत कार्बन डायऑक्साईड जर वाढत असेल तर फोटोसिंथेसिसचा रेट वाढतो. ओके, आणि यानंतर जर गेला तर रेट कमी होतो. ओके, सो देर इज ऑल्सो दी ऑप्टिमम value. Okay, so after optimum value, if you increase the same parameter, so rate will not going to increase. It's a rate limiting factor under the normal temperature and the pressure. So normal temperature and in the normal pressure, if you say, the carbon dioxide is act as a rate limiting factor. So we are going to discuss about the rate limiting factor. What is it? So temperature, if you say, the temperature is also the rate limiting factor, or the it is affecting the photosynthesis. How the photosynthesis also need a suitable temperature. Okay, so it also need the suitable temperature, like other physiological processes in our body or in a body of a plant. We know that there is the different physiological processes which is completely depending upon the temperature. Okay, so the temperature is also needed for the photosynthesis. uh, with increase in a temperature, the rate of photosynthesis has also increased. So the rate of photosynthesis is directly proportional to the temperature. Okay, optimum temperature for photosynthesis is 25 to 30 degrees Celsius. Okay, but if you see Opensia, so it is a desert plant. Okay, we know that it is a desert plant. It shows the photosynthesis at 55 degrees Celsius. So if you see the 55 degrees Celsius, the photosynthesis is working. So it is called as a maximum temperature. Okay, so what is meant by minimum, guys? So minimum means where the process starts. Okay, and maximum means where the process ends or stops due to the high temperature. Okay, so this is called as maximum and minimum parameters. Okay, and optimum means the, let's say, in the form of graph, if you say, okay, for example, this is your graph. So this point showing the maximum value, this point showing the minimum value, and this is your optimum value. Okay, so this is the actual difference between maximum, minimum, and optimum. Water increase in a water content of a leaf result in increase in a photosynthesis. Okay, water content of a leaf, increase in water content of a leaf result in increase in a photosynthesis. So water is, you can say, directly proportional to the rate of photosynthesis. Okay, so the factor affecting, one more factor. So these are the external factors, these are not present inside of a plant. The temperature, water, and the light, and carbon dioxide, these are the environmental factor. But we have some factor which is living inside of a plant, that is internal factor. Photosynthesis is carried out by chlorophyll, so the rate of photosynthesis is proportional to the quantity of a chlorophyll, that is the number of chlorophyll. Okay, so the more number of chlorophyll will definitely increase the rate of photosynthesis. That's why the number of chlorophyll or the quantity of chlorophyll is directly proportional to the photosynthesis. Okay, product of photosynthesis is sugar. Accumulation of sugar in a cell slow down the process, because the more sugar inside of a cell will give the bulkiness to the cell, and this bulkiness do not efficiently, I mean, in this bulky cell do not efficiently work with the photosynthesis, because the light which is coming inside also affected due to the these sugars. Okay, so the thickness of epidermis and the cuticle of a leaf also affect the rate of photosynthesis. Okay, so these are the for example, let's cut the leaf like this. Okay, for example, this is leaf, we just cut it. So these two layers, the outer and the, so the upper and the lower, so these two layers, or these two cells are the cuticles and the epidermis of your leaf. Okay, so these are the actually epidermis of leaf. So the thickness increases, so the light is not going to penetrate much more. Next one, the distribution of a stomata in a leaf. So the stomata is present actually the lower part always. Okay, so the distribution should be a very good. The distribution is not in a perfect way, so it will also affect the level of carbon dioxide going to be decreased due to the not good distribution of stomata, and that's why it will result into the low concentration of carbon dioxide inside of a leaf, and it will result into the low rate of photosynthesis. So the distribution is correct. So the rate of photosynthesis is going to be increased. It is also the factor. Okay. So the Blackman's law of limiting factor. So this is the very important thing, guys. You will definitely see in the examination Blackman's law of limiting factor. So what is the law? Let's say the law says that the Blackman's law of limiting factor state that when a process is condensed as to its rapidity by the by the number of a separate factor. The rate of a process is controlled by the pace of the slowest factor. Okay, so what is meant by the law? Like, let's see, guys. Let's take an example. So we are going to take an example, the photosynthesis. Okay, so suppose the plant photosynthesis is now going on with a fixed speed. Okay, and speed किती आहे? So it is now depending upon the intensity of light and the carbon dioxide. Okay, so these two values are now the in a perfect way. Okay, so these two values are very good for the 10 mg CO2 fixation per hour, for example. Okay, so I am writing here in the slide also. So for the 10 mg carbon dioxide per hour fixation, the requirement of carbon dioxide and the requirement of light is ready. But if you increase the carbon dioxide further, so the increase in carbon dioxide will definitely result into the increase in photosynthesis. Okay, but after the more increase in carbon dioxide, if you see the large amount of carbon dioxide you are increasing in environment, but the rate is not going to be very high, and that's why if you see the graph of this the condition, it will look like this. ग्राफ कसा दिसणार रे? तर कार्बन डायऑक्साइड वाढवला तुम्ही हळूहळू वाढतोय, त्यानंतर किती कार्बन डायऑक्साइड वाढवला, सो हियर इज युअर कार्बन डायऑक्साइड. Okay, so and this is the intensity of light. Okay, so if you increase the carbon dioxide, the rate is going to be an increase, but after the single point, the increase in carbon dioxide will not going to increase in the rate of your photosynthesis. Okay, so in this situation actually your light is becoming the this intensity of light is becoming the rate limiting factor or the limiting factor. Okay, so this is called as a limiting factor. आता तुम्ही म्हणाल की सर लाईट कमी पडते, आपण लाईट वाढवूया. तुम्ही जर लाईट वाढवली तर परत एकदा वाढणार आहे, पण त्यानंतर तुम्हाला दिसेल की फॉर एक्झाम्पल हा रेट आहे, लाईट नाही, रेट ऑफ फोटोसिंथेसिस. ओके, मग तुम्ही आता इथे CO2 घेऊ नका, लाईट घ्या. लाईट वाढवल्यानंतर एका लिमिट नंतर परत फोटोसिंथेसिस स्टॉप होणार आहे. मग किती लाईट वाढवली तर तुम्हाला फोटोसिंथेसिस वाढताना दिसणार नाही. या पॉइंटला जर तुम्ही पाहाल तर परत कोणीतरी दुसरा एखादा फॅक्टर इज नाउ गोइंग, दिस एक्स फॅक्टर इज गोइंग टू बी बिकम द लिमिटिंग फॅक्टर. कोण असेल? मग फॉर एक्झाम्पल CO2 पण भरपूर आहे, लाईट पण वाढवली, मग पाणी कमी पडेल किंवा दुसऱ्या कोणत्या गोष्टी कमी पडतील, कोणी ना कोणी लेट लिमिटिंग फॅक्टर म्हणून काम करणार आहे. आणि जो पण लिमिटिंग फॅक्टर असतो ना, तो नेहमी लोवेस्ट अमाउंट मध्ये. हा जो स्लोवेस्ट फॅक्टर आहे ना, इट इज ऑलवेज प्रेझेंट इन अ लोवेस्ट अमाउंट. ओके, हा लोवेस्ट अमाउंट मध्ये प्रेझेंट असतो, कधी पण लक्षात ठेवायचं. जो स्लोवेस्ट फॅक्टर असणार आहे, सो दॅट स्लोवेस्ट फॅक्टर इज ऑलवेज प्रेझेंट इन अ लोवेस्ट अमाउंट. ओके, So the if you increase the carbon dioxide, the light will become the rate limiting factor or the limiting factor. If you increase the carbon dioxide and light both, so you are the third factor, anything the maybe the water, maybe the let's say the temperature. Okay, so this any other factor is becoming the limiting factor. Okay, and Blackman, this is the Blackman's law. Okay, Blackman म्हणतो की जर तुम्ही एखाद्या प्रोसेसचा रेट वाढवण्यासाठी एखादा पॅरामीटर जर इन्क्रीज केला तर एका लिमिट नंतर तो किती इन्क्रीज केला तरी रेट मात्र तेवढाच राहतो, स्पीड मात्र तेवढाच राहतो. का तर दुसरा कोणीतरी पॅरामीटर लिमिटिंग फॅक्टर म्हणून काम करत असतो. मग तुम्ही जर दुसऱ्याला वाढवला तर तिसरा कोणीतरी लिमिटिंग फॅक्टर म्हणून काम करेल. मग तुम्ही दुसरा आणि तिसरा दोन्ही वाढवले तर पहिला लिमिटिंग फॅक्टर म्हणून काम करेल. कुणी ना कुणी तिथे ऍट अ टाइम सगळेच्या सगळे पॅरामीटर तुमचे काम करणार नाहीत. कुणी ना कुणी रेट लिमिटिंग फॅक्टर म्हणून काम करतो आणि म्हणून तुमचा जो त्या प्रोसेसचा स्पीड आहे किंवा तो प्रोसेसची रॅपिडिटी आहे, सो इट विल बिकम दी स्टेबल. ओके, आणि मग ग्राफ कसा जातो तुमचा, तर हा ग्राफ जो आहे तर हा ग्राफ सगळ्यांनी लक्षात ठेवायचा. So the rate of your process is going to be an increase, and after the point where the one become the limiting factor, so it will bend and it will become the flat. Okay, so this is the graph. So thank you very much guys. आज संध्याकाळी 7 वाजता आपण फोटोसिंथेसिस नर्वस सिस्टम मध्ये ब्रेन ला सुरुवात करणार आहोत. Okay, so today we are going to discuss about the brain, and tomorrow उद्या आपण या फोटोसिंथेसिस बद्दलच्या बाकीच्या काय प्रोसेस आपल्या राहिलेल्या आहेत, लाईट रिएक्शन्स वगैरे आपल्याला दिवाळीमुळे काही गोष्टींमध्ये सुटलेल्या आहेत, तर त्या गोष्टी आपण उद्यापासून स्टार्ट करणार आहोत मित्रांनो. ठीक आहे, तर आज संध्याकाळी 7 वाजता आपण ब्रेन बद्दल माहिती घेणार आहेत. सगळ्यांनी वेळेवर यायचंय आणि सगळ्यांनी लाईव्ह यायचंय, कोणी दांड्या मारू नका. Okay, so thank you very much. If you like my lecture, please hit on the like button, and this is the Telegram channel for you guys. It is for the MCQs and the notes. Okay, so first subscribe and click on the bell icon. So this is Crack Maharashtra CET for 2021. Okay, 2021 च्या विद्यार्थ्यांसाठी हे CET ची बॅच आहे, लक्षात ठेवायचं सगळ्यांनी. आणि सबस्क्राइब करून ठेवायचा चॅनल आणि बेल आयकॉन वरती क्लिक करायचा. टेलिग्राम वरती तुम्हाला नोट्स आणि एमसीक्यूज अवेलेबल आहेत विथ डेली अपडेट्स. डेली अपडेट्स म्हणजे कोणते लेक्चर कधी असणार आहेत याबद्दलची इन्फॉर्मेशन इथे आहे. प्लीज ट्राय टू अटेंड दी लाईव्ह लेक्चर्स. लाईव्ह लेक्चर अटेंड करा रे मित्रांनो, तुम्हाला काही गोष्टी कळतात, नंतर समजत नाहीत आम्ही नसल्यावरती. ओके, सो थँक यू व्हेरी मच.